In prokaryotic and eukaryotic cells, we are going to discuss about the principles of the cell theory. Then we are going to see the details of the structures of prokaryotic and eukaryotic cells. And then illustrate and compare the structures of prokaryotic and eukaryotic cells. Now let's look at the cell theory. We have to memorize. Students should memorize these theories so that when question gives you a statement, you know that whether it is a cell theory or not. Okay, related to the cell theory or not. Or if you were asked to explain cell theory. According to the theory, all organisms are composed of one or more cells. So for those organisms with only one cell, we call them unicellular. And then we have more than one cell is multicellular. The second theory is saying new cells are produced from the earlier cells through cell division. Mitosis, meiosis, binary fission. Then cell is a unit in which all metabolic processes take place. So a cell is a basic unit that every important thing occurs in a cell. And each cell, the last one, each cell carries heritable or genetic information. Mesti ada, So this genetic information can be passed on to the daughter cells during cell division. So it is also related. These are diagrams about prokaryotic cells and eukaryotic cells. So based on this diagram, we know that eukaryotic cells can be divided into animal cell and plant cell. What are prokaryotic cells? Prokaryotic cells are cells that have primitive nucleus. So when we have to describe what is primitive nucleus, it is a materials when the genetic materials is not enclosed in any membrane in the cell. Because primitive pro means primitive, early, and karyo is nuclear. So it is they are cells that have primitive nucleus, meaning that their nucleus does not look like our typical nucleus. Very primitive because they don't have nuclear membrane. Make sure that you are able to draw a typical structure of prokaryotic cell. So when we have to draw, there's always don't forget about the capsule. First, mesti ada bentuk. And then we have the plasma membrane. The plasma membrane. And then there is a cell wall surrounding the plasma membrane. And then we have another layer, which is called capsule. Right. This is the cell wall, okay? Sama nampak And then apa yang penting di dalam prokaryotic cells? There is no nucleus, but the genetic materials will be lying in the cytoplasm, okay, scattered in the cytoplasm. So we can just draw it. This is called circular DNA. Circular DNA because apa beza circular dengan kita punya DNA? Our DNA is called linear. So can you differentiate between circular and linear? Somebody?
Ini macam straight line, madam.
Yeah, straight line. So kalau kita kena, if we have to describe the difference between a linear and a circular, what makes them different? You see, linear is like this. Circular is like this. So what's the difference then?
The DNA with two ends, while circular DNA with no ends.
Okay, the linear DNA, it has the begin atau dia ada dua ends kan? Sorry. But circular DNA, the end fused together. So there is no end. Tu beza dia. Maknanya DNA kita meaning that our DNA, it starts here and it ends here. But in bacteria, the DNA starts ends where it starts. Tempat yang sama Itu adalah circular DNA. Sebab tu bila lukis ni dia akan ada, sepatutnya akan ada, takkan ada ends dia akan bertembung ujungnya. Next, the presence of flagellum, and then here is plasmid. Plasmid is another type of DNA. So prokaryote basically have two kinds of genetic materials. One is the DNA and another one is called plasmid. Plasmid is also DNA but it is smaller constituent ataupun lebih size dia. The size is smaller than the DNA, main DNA itself. Okay, ada pilus. Kalau plural kita sebut pili. Then we have mesosome. If you look in the structure, the only organelles that we can find is the ribosome. Okay, ribosome. There is no nucleus, there is no mitochondria, what else? Chloroplast or Golgi body and everything. Tak ada. Sebab tu nanti bila kita bezakan, those are a very distinctive features between prokaryote and eukaryote. Prokaryotic cell. The key concept is prokaryotic cell lack membrane enclosed organelles. Ada juga kita gunakan istilah membrane bound. Membrane bound organelles. Membrane enclosed organelle. Tidak ada organelle yang bermembrane. Okay, ribosome is an organelle but it does not have a membrane. It is a folded polynucleic acid. Ribosome. All bacteria contain cytoplasm, ribosomes, a plasma membrane, and nucleoid. So nucleoid is where is the region where we can find their DNA. And almost all bacteria have cell walls. Generally small in size. Small in size meaning that they are very micro. We can only observe them using microscope or magnified a few hundred times. They has no membrane enclosed organelles. Contain small size ribosome for protein synthesis. So they also have ribosome, but their size of ribosome is not similar to us. Capsule presence in some bacteria, which is a jelly-like or sticky layer of polysaccharide, polysaccharide made up of polysaccharide and protein. The function of capsule is to protect against dehydration or protect them from attacks by host immune system. Host ke sini, imagine yourself, because we have bacteria in our intestinal tract, right? Such as E. coli. living in our intestine. So we are the host. So our body has a very capable immune or defense system. We have the hydrolytic acid. We have a few more enzymes that can surely digest the bacteria, but they are protected by this capsule. That was why they can E. coli, such as E. coli, bacteria such as E. coli, can assimilate or living inside our body. Next, we have the cell wall. Sorry, not we. The prokaryotic cell has cell wall that composed of peptidoglycan. You have to memorize this composition because the composition of cell wall between prokaryote and eukaryote are very different. Their cell wall composed of peptidoglycan. Dah ingat dah? The function of the cell wall is to protect the cell and maintain its shape. So they can prevent any excessive water uptake, because if they absorb water more than they should, they might get bloated or burst, pecah, hemolysis, like our red blood cell. But because there is cell wall surrounding their structure, then they can hold or they can prevent the excessive water uptake. So what is peptidoglycan? Also stated in the lecture, in the notes. You can read them. What I want to highlight is look at the difference between these two type of cell walls of bacteria or prokaryotes, bacteria. To be more specific, these two types of cell wall present in bacteria. We have a gram-positive bacteria and gram-negative bacteria. So their composition of cell wall is different between this gram-positive and gram-negative. Okay, why? First, in gram-positive bacteria, the peptidoglycan and the plasma membrane, they are uh, attached or embedded together side by side, while with the peptidoglycan as a thick layer. Okay, dekat luar peptidoglycan thick layer. While in gram-negative cell wall, the peptidoglycan is a thin layer, thin layer. Thin layer, then the peptidoglycan is entrapped in between the outer membrane. The next structure is the pilus. Pilus is a structure that helps in conjugation. Okay, shorter than flagellum. Okay, pilus shorter than flagellum but longer than the fimbriae or fimbriae. Jadi bila conjugation is a way of reproduction, bacteria reproduction or microorganism reproduction. Or they can also they also use the pilus to communicate with another bacteria. Fimbria is a short hair-like appendages and numerous than pili. It helps bacteria to adhere to the substrate or to other cells such as rocks, atas batu. Then cytoplasm is the same with any other cells. All cells contain cytoplasm, surrounded by plasma membrane, encased with a rigid cell wall. The DNA of prokaryotic cells located in the nucleus. The DNA is circular and not associated with histone protein, right? Circular and not associated with histone protein. Next, mesosome. Mesosome is the replacement or for eukaryote, we have the the mitochondria to generate ATP or energy. But for prokaryotic, since they don't have the nucleus membrane-bound organelles, they don't have the mitochondria. The mesosome is the site where they respire or undergo cell respiration. Mesosome is actually infolding, okay, lipatan, infolding of the plasma membrane yang berlipat ke dalam. So it becomes the site for cellular respiration. While flagellum or plural flagella, in prokaryotic cells, the flagellum lack 9 plus 1 microtubule arrangement. Okay, apa dia microtubule 9 plus 9 plus 1 pula. 9 plus 2, apa dia? Boleh baca dalam nota ataupun dalam buku rujukan ataupun mana-mana rujukan dalam internet dia ada nyatakan. Susunan dia 9 campur 2. The flagellum is made up of protein flagellin, has long and thread-like structures protruding from the surface and move in rotation. Okay, can you name one cell in animals that has flagellum? In animal or in eukaryotic cell? Siapa saya nak panggil? Najatul Sarafinas. Can you identify any cell in our body that contain flagellum? Or animal in general?
Sperm.
Sperm, very good. Sperm is the only cell in animal that has flagellum. So kalau dia kata kat dalam ni tadi nota dia kata dalam prokaryotik flagellum dia lack of 9 plus 2. Maknanya dalam eukaryotik dia punya susunan mikrotubul adalah 9 campur 2. The flagellum in prokaryote is used for locomotion. Locomotion maksudnya mobility or movement and feeding. Feeding makalah, tak tahu kan. Next is plasmid. Plasmid is a small circular DNA. So, kalau kena terangkan mesti ada perkataan small circular DNA. Dia small. Nampak tak beza ni? DNA yang ini dengan plasmid. Right? Jadi dia kecil. Only contain a few genes. Maksudnya hanya ada beberapa gene sahaja yang ada dalam plasmid. Kita akan belajar dengan lebih lanjut lah apa fungsi plasmid nanti dalam bab 6 contoh. Eukaryotic cells have true nucleus. True nucleus. U means true. True nucleus because the DNA, the genetic materials are enclosed in a nuclear membrane. Okay, nuclear membrane. The typical structure of animal cell, because eukaryotic includes animal and plant cell. So kalau kena lukis, typical structure ada dua. Satu, typical structure under light compound microscope. Kalau under light compound microscope, kita hanya akan nampak cell surrounded by plasma membrane, and then a nucleus, nuclear membrane. Sometimes if we are lucky, kalau kita tengok dalam tumbuhan contohnya, kalau dia tumbuhan tu sel meristem, sedang aktif membahagi, kita mungkin akan nampak nucleolus lah. Okay. Then kita boleh nampak mitochondria. The rest kita tak berapa nampak Golgi body, apa lagi, rough ER, smooth ER, yang ni kita tak berapa nampak lah bawah light compound microscope. Okay, tapi ni adalah typical structure yang kita akan nampak kalau kita nak cerita tentang animal cell, selalunya bawah electron microscope. Okay, what are they? We have smooth endoplasmic reticulum. Endoplasmic reticulum, we have the smooth. Then we have the rough endoplasmic reticulum here, studded with the ribosome. We have Golgi apparatus. We have ribosome, then lysosome. Do not forget the mitochondria. Then we have the nucleus, of course. What else? What did I miss? The ribosome. Ribosome already. Okay, nucleus. So yang dinamakan organelle adalah this one, nucleus, endoplasmic reticulum here, either smooth or rough, mitochondria. Vacuole also can be included as organelles. Tapi dalam animal cell vacuole tak nyata sangat Vacuole tak nyata sangat. Then ribosome also will be discussed, Golgi apparatus ataupun Golgi body. Okay, nucleolus is in the nucleus, so we are going to discuss about nucleus, not the nucleolus per se. Okay, next. Ni boleh baca. Saya tahu awak dah biasa dengan ni. Untuk plant cell, kita ada tambahan cell wall. Lepas tu apa lagi? Okay, central vacuole, then chloroplast. Yang lain sama. Generally, eukaryotic cells are bigger than prokaryotic, has various membrane-bound organelles. Next. Tak ada apa Cell wall. For eukaryotic cells ada tiga jenis hidupan Kita ada plant, fungi and also animals. So, nanti semester 2 baru kita akan tahu ni plant and animal is under the same kingdom, fungi is a different kingdom. So the composition is different between these three type of cell. Flagella, okay, tadi dah mention, so saya teruskan. DNA as genetic material found in the nucleus, surrounded by the nuclear membrane. So the DNA is linear and associated with histone protein. This is a very important description that we have to remember. Eukaryotic cells are supported by cytoskeleton. Maksud dia rangka dalam dan rangka luar. Kalau kita sebagai manusia as a whole, kita ada skeleton, tulang, kita ada cartilage. Okay, tapi untuk satu sel saja, macam mana dia boleh maintain the shape. Okay. Maintain the shape. Walaupun animal cell kan dia tak ada dinding sel kan. Tapi macam mana dia boleh maintain that shape because of the cytoskeleton. The function is to give mechanical support and maintain the shape of cell. Now, as you can see, you dah baca pun comparison between prokaryotic and eukaryotic cell. Is there anything that you want to ask before I continue with the next subtopic? I'm not going to read out everything because you can read it yourself kan. Cuma saya nak tahu ada tak yang you tak faham. Okay, saya dah jelaskan tadi ni semua ada. Okay, small ribosome, prokaryotic cell, the small ribosome is 70 S type. While eukaryotic cell, we have a larger ribosome, 80 S type. Prokaryotic cell divides by binary fission. They do not undergo mitosis or meiosis. But for eukaryotic cells, our cell division is by mitosis or meiosis, or both. Okay. Membrane, the cell wall, and the plasma membrane.